I too would welcome you to this sixth meeting of the Australian Academy of Health and Medical Science, and for the small numbers of us here to Royal Prince Alfred Hospital, where we're providing a local audience. And it's wonderful that so many people can join us from around Australia. We have a particular treat in store this morning with a series of excellent presentations on the role of precision medicine. And I would first like to introduce Dr. Judith Cho. Professor Cho is the director of the Charles Bronfen Institute for Personalised Medicine and the Dean of Translational Genetics and the Ward Coleman Chair for Translational Genetics at the Icahn School of Medicine at Mount Sinai. Since 2015, Dr. Cho has led the Institute, which includes the School of Medicine's major biobank, BioMe, which represents one of the most diverse biobanks in the world, and sequencing results from that underscore the enormous potential of genetics as a strategy in clinical care. But as with many of us, with the SARS-CoV-2 pandemic, Professor Cho has played a lead role in switching some of her research to COVID-19, organising the Mount Sinai School of Medicine's efforts in genetics and serological studies, anticipating, as we all do, a successful vaccine development in the near-term future. Her own outstanding research focuses on integrating genetics and single-cell genomics to develop new therapies in inflammatory bowel disease. Single-cell technologies are providing an unprecedented new look at human biology, and the next phase will integrate targets for which existing banks of tissues are available to contribute to the research to find new therapies for this condition. We now have the opportunity to hear Professor Cho from New York.
So it's a genuine pleasure to be a part of this meeting, unfortunately from halfway across the world, but I'm happy to share with you my thoughts on precision medicine research and how to move from that to improving clinical care. So just a little bit of background about myself. I direct the Bronfen Institute for Personalized Medicine at Mount Sinai in New York. I also have traditionally studied the genetics of inflammatory bowel disease. I used to practice gastroenterology. I no longer practice, so just as a caveat in terms of how we can use research to improve clinical care, I want to put that caveat in there. But the three examples that I wanted to talk to you about are first, how the precise genetic understanding leads to a precision or a capacity to treat patients. Show you a couple of slides about imaging in healthcare systems. These represent untapped treasures of research insight that can improve clinical care. And in the final section, I'll be talking about timing of health and disease prediction and scaling research to improve clinical care in large health systems. So any kind of talk where we try to be a little aspirational, we're trying to look into the future, it can be challenging. You want to skate to where the puck is going, not to where it has been, according to Wayne Gretzky. And one of my favorite quotes from Bill Gates is, we always tend to overestimate the change that will occur in the next couple of years and underestimate the change that will occur in the next 10. So with these caveats, I think that Mount Sinai really has been one of the leaders in trying to apply precision medicine genetics in the improved care of patients. So I'm happy to share with you what we're doing at Mount Sinai. So the BioMe Biobank at Mount Sinai is an electronic health record linked biobank where patients consent to prospective and retrospective mining of their electronic health record. We also obtain questionnaire data. Currently, we have over 60,000 participants where we have both DNA and plasma. And one of the strengths of having a biobank based in the heart of New York City is that the participants are highly diverse from a population perspective. Approximately 40% Hispanic, 35% European ancestry, and 25% African American. And this enormous genetic diversity is important not just from a social justice perspective, but also improves our science. In addition, the patients consent in BioMe to nested studies. So if they want to do side studies off of a subset of particular patients. And a major advance that we made in about 2018 was that if we took the DNA information, sequenced them, and found actionable findings, did the patients want us to contact them? And overwhelmingly, patients have wanted to be recontacted if there's an actionable genetic variant. So in this process of going from research to clinical care, I think we have some of the important niduses for making that transition of going from research to clinical care broad scale across the health system. So the basis, a substantial basis for BioMe is gene-based precision and genetics first. So for years, people like myself who were geneticists doing genetic discovery would collect a bunch of patients with a particular phenotype of interest, in my case, inflammatory bowel disease, and then perform genotyping to identify which regions in the genome are associated with the phenotype of interest in order to do gene discovery. But the flip side with these biobank-based collections is that we've had many years now of genetic discovery. Now the goal is to flip that paradigm and to take certain genes of interest and then systematically go backwards and understand what's the nature of the phenotypes that are correlated given a patient who has a particular genetic variant. And so a couple of examples of this is that you can create precision in our understanding of a very common trait that patients often present with, congestive heart failure. And in medical school, we're often taught about of horses and zebras. If you hear hoofbeats, think horses, not zebras. And congestive heart failure is a very common diagnosis. But occasionally you'll have a subset, a particular type of congestive heart failure, which confers a particular different type of traditional therapies that would be used. And an example of this is our discovery that we reported last year in JAMA of the effects throughout a health system of individuals who carry these mutations in this gene called TTR or transthyretin, which causes a particular type of cardiomyopathy with this very specific type of treatment. And so the particular variant is this valine 122I in transthyretin, and it's associated with congestive heart failure in 2 independent biobanks. So this represented a very nice collaboration between the Sinai team, Rondo, Girish Nadkarni, and Kumar Chaudhary, as well as the Penn team led by Dan Rader and Scott Damrar. And so we systematically looked at patients, looked at their genetic information, and said, let's take a gene-first approach and understand what's the likelihood that you're going to have problems because you carry these particular mutations. And individuals who carry those mutations had a 1.6 to 1.8 odds ratio for heart failure. And this particular variant is particularly prevalent in African Americans as well as Hispanic populations. And the reason why it's so important to make this diagnosis from a genetic perspective is that recently approved is the drug tafamidis, which is a small molecule which stabilizes TTR, thereby preventing the development of this heart failure. So again, this type of very exquisite precision in the general umbrella of congestive heart failure is terribly important because we may be able to identify more specific treatments for individual patients. And we can take this one step farther, is that BioMe represents 60,000 or about 1% of the Mount Sinai health system. Can we then, even without the presence of the DNA information, develop predictive models to proactively push information that suggest this particular variant, these particular clinical profiles suggest that maybe you may be more likely to carry the TTR mutation, so consider genetic testing. So there's different ways that you can think about these interactions. So the TTR mutations, as I alluded to earlier, are more prevalent in African populations, and there is a particular value to African populations. So both through BioMe, as well as our work in inflammatory bowel disease, we're particularly highlighting the collections of African American cases for the obvious reason that we are all Africans. All of human history evolved from the continent of Africa. And in terms of the particular value of these African populations, it has the greatest genetic diversity of all populations. The polygenic risk score, which I'll elaborate in a couple of slides, unfortunately don't translate well across populations. And one of the particular features of African populations is they have the shortest linkage equilibrium blocks. And we have some data that I won't share today, but that suggests that combining African with European populations are actually improves predictions for all populations. So there's a particular value to emphasizing the African collections. And then the final point I'll make in the genetics section of this talk is that I've been serving on the organizing committee of the ICDCA, which is the International Common Disease Alliance, which has the lofty goals of connecting these genetic maps to mechanisms of disease, to the development of medications. This is the brainchild of Eric Lander, and also being led by Cecilia Lindgren from Oxford. And there's a couple of particular points. We've talked about biobanks, and there's value in our biobank to collecting DNA and plasma specimens, but there's also value for patient registries where you can consider many different ways where patients can actually report information to registries that are going to be valuable. And the other area of ICDA that I've been very active in is this last part of the medicines part, where we're trying to think about how we can, in a ethical and responsible way, make recommendations regarding the use of polygenic risk scores, which are these genome-wide common variant prediction for common diseases. It's currently unproven benefit in clinical care. Most of the kind of genetic evaluation in clinics now are focused on these rare variants of high effects. But I think it's probably pretty likely that polygenic risk scores may be attempted to be applied in health systems broadly. So the second vignette I'd like to talk about is imaging within health systems. This is an untapped treasure of information. And part of this reflects the changes in medical care, increasingly non-invasive imaging through capsules, CT angiographies, fiber scans. There's a vast amount of information that is being developed in addition to the most important sources of imaging data, radiology, and pathology. And in terms of radiology applications in healthcare systems, this is happening now at Mount Sinai and many different other health systems. And you can think of this generally as two different data types. One is using natural language processing, which can abstract SNOMED terms from these text reports of radiology or pathology. Or you can actually go to the primary image analyses themselves. If you scrub all of the personal identifying information, you can take that raw image data to classify things and combine this more generally and get information beyond just the report itself. So just to extend one more point in terms of pathology and the single cell revolution, this is a paper that we published last year where we did single cell transcriptome analysis off of Crohn's disease biopsies, validated some of our key findings, most importantly here by protein analysis, the PDPN gene, which is a marker of an activated fibroblast marker, together with the CD68 macrophage marker, present in particular subsets of patients with Crohn's disease. And in particular, what we go on to show in this paper is that the differentially expressed genes by single cell sequencing can then be projected onto larger bulk RNA-seq cohorts with treatment response data. More generally in pathology, what is happening at the clinical level is you're having digitization of the pathology slides, which again forms an enormously rich reservoir of information. And even beyond just the image data themselves, the paraffin blocks are stored for years in the pathology departments and serves as an enormously rich resource for the development of kind of sliced scrolls, which can then be used for additional analyses. So perhaps one of the biggest challenges in biomedical research and in healthcare systems is the fact that so much of human health and disease has substantial time course considerations. And understanding this and being able to follow these key factors over time is increasingly important. One can think of, from a health system population-based perspective, the attempt to prevent disease, which is really one of the predominant cancer paradigms, one of the ways from a public health perspective, we want to control the impact of cancers. Another important factor is that there's an explosion of the capacity of us to measure protein biomarkers as a measure of potentially new biomarkers that can be used for both disease prediction as well as treatment course. So in terms of these timeline or lifespan considerations, just to give a couple of examples in inflammatory bowel disease, this was from a very nice review article published by Holm Elig a few years ago in Gastroenterology. So inflammatory bowel disease has, for most cases, if you look at that bottom area of the Oxford IBD cohort, you can see that the peak age of IBD onset is between 15 and 30 years of age. And that's kind of the usual or more polygenic forms of inflammatory bowel disease. But intriguingly, what we've known for years is there are some people that present with intestinal inflammation at a very early onset, typically defined as being diagnosed less than 10 years of age. There are some cases where patients present at birth with a neonatal diagnosis of intestinal inflammation, and autosomal recessive mutations along the IL-10 pathway contribute to this. And one example that I just want to show you is this gene called LRBA, which in autosomal recessive forms have been associated with autoimmunity in children. But in our BioMe data, we identified through a whole exome sequencing heterozygous carriage associated with a later onset African-American IBD. So LRBA blocks shuttling of the surface protein CTLA4 to endolysosomes. And so we showed that the LRBA expression of the carriers of this African predominant variant, valine 737 isoleucine, has decreased LRBA expression, as well as decreased CTLA4 expression, which can be partially rescued by chloroquine, as shown in the red box whisker plots in both carriers as well as wild-type individuals. I bring this point up because chloroquine has had some play in terms of potential efficacy in COVID, but where it's clearly established to be efficacious is in improving survival in a related inflammatory disease called systemic lupus erythematosus. And to the point even that it's so efficacious that clinical trials to actually prevent the onset of lupus have actually been proposed. And so if you look at the timeline of pathogenicity with autoimmune diseases, we know that there's a substantial germline genetic architecture that can be modified over the course of years. In most cases of these autoimmune diseases, you don't actually present, except in the very early onset cases, immediately. And you have this period of years where you have an altered immune system as manifested by these autoantibodies. And then depending upon the type of organ system that is the main source of the structural damage, a target of the structural damage, the patients will present with disease. And one of the major challenges in the inflammatory bowel disease field is identifying the optimal timing of interventions. And our goal increasingly is to intervene with major medications earlier on a disease course. Then switching to cancer, the other major factor, this is the, in terms of the cancers, are the major actionable genes that contribute to precision medicine and the desire to actually catch these mutations. And so this is from the ACMG list of 59 actionable autosomal dominant variants. And you can see that these actionable autosomal variants that confer risk for cancer genes can present either as in childhood or adulthood. And so these are the kinds of variants that if we know patients carry them, there's going to be specific screening action to try to prevent the onset of cancer in these individuals. So in terms of thinking about how you manage cancer from a population perspective, one of the factors in terms of electronic health records in the United States, again, don't know exactly how it is in Australia, but I suspect many of these challenges that we have with electronic health records are similar to what you have in Australia. So the EHRs, or electronic health records, reflect the patterns of healthcare and healthcare delivery, which is often fragmented. And you can think of the data that exists in the EHRs as being structured data, such as laboratory reports, ICD diagnoses, as well as textual reports. One of the major projects that we're going to be undertaking this year and next is the movement of our entire electronic health records at Mount Sinai, constituting 4 to 6 million patients, into this new OMOP model, or common data model. And then a couple of challenges are that medication use has traditionally been very challenging to mine. It's again unclear kind of the quality of the medication use to try to track patients over time versus relying on the claims databases which reflect the payment for these medications. And finally, apropos of these monogenic forms of cancer, family histories are very inconsistently recorded and acted upon. So I think one major development we're going to try to work on at Mount Sinai is to systematically work on data systems to capture family history, especially in these cases of familial cancers. So electronic health records have their limitations, and I think one of the solutions will be digital health, patient-based digital health, wearables, and understanding the very different rhythms and ages of human health and disease. You can think of progressive diseases like coronary artery disease, the inevitable progression towards chronic kidney disease in some patients, and cancers. You can think of the major degenerative diseases such as neurodegenerative diseases, as well as joint diseases. You can think of episodic diseases like inflammatory bowel disease. And so I think digital health represents an important way to maintain patient engagement, which is essential, and another way of getting actually probably the best family history you can possibly get. And then when you think about doing machine learning and integrating these digital health data streams, one of the key things when you design these studies is to think about outcomes: survival, cancer rates, mobility, hospitalizations. These are all things that you can measure quite well with these digital health approaches. At Sinai, we, led by Aniwa Owusu-Obeng of PharmD, we are piloting a volunteer-based program for individuals to undergo pharmacogenetic testing starting in December. The value of this are in certain classes of drugs, such as platelet inhibitors, antidepressants, antibodies to biologic agents, such as anti-TNF. And the hope is by prospectively testing these individuals will have reduced side effects and enhanced drug efficacy. Some of the things that we're hoped to work out through these pilot studies is understanding the particular efficiencies of the pharmacy department versus the physician, focusing on these pharmacogenetic studies, as well as establishing the clinical decision support computing systems so that, okay, you may not have any unfavorable pharmacogenetic drug genetic pairings now, but it's possible that a future healthcare provider may order this in the future. So how do you create computer systems that can anticipate those needs? So in conclusion, you know, if we think about a wish list, if we try to think big about what we want biomedical research to accomplish in these academic healthcare systems, we have to think about the financial, clinical, and socioeconomic constraints and limitations that we have, and how can we work to minimize diagnostic delays and delays in providing the most effective treatments. It's inevitable that these apps and these wearables are going to be a future part of future healthcare. And it's important from a social justice perspective to make sure that there is no digital divide. We want to be able to enhance communication with our patients, and apps are just going to proliferate in healthcare as they have in other parts of our economy and our lives. Ideally, I think cancer has led the way in precision medicine. And ideally, we across the board, we can adopt the cancer paradigm with more broad-scale research involvement. And then also, can we use the electronic health record? Can we use the data streams to apply more routine use of machine learning in clinical care? We've talked about imaging and possibly omics as well through these polygenic risk scores and expression studies through RNA sequencing. And finally, in terms of medications, if you think more broadly, if you think more holistically across a health system, you can potentially accelerate and enhance therapy and repurpose drugs more efficiently, and also perform genomically informed clinical trials. So our goal really is to scale crosstalk between research and clinical, and really erase that divide so that we can accelerate benefit to patients in a better, faster, and cheaper way. And so one of the best examples of this represents a joint effort between Alex Charney and Girish Nadkarni here at Mount Sinai in response to a real emergency, obviously the pandemic. And in response to the pandemic, which hit New York quite heavily in March, they on the spot developed a Mount Sinai COVID Intelligence Center where they combined all these different data streams of things that were just traditionally clinical care, quality of care, and created a data information platform team that could address the immediate needs. Then they were substantial immediate needs to address the COVID emergency. And through that, they were able to very quickly on the spot identify certain medication trends, such as the early use of anticoagulations to prevent the clotting complications that we see with COVID, to actually improve survival in patients in real time to help patients really at the heart and the teeth of the pandemic. And so subsequent to that, luckily, thankfully, the rates of COVID over the summer dropped a bit. And so they've morphed the Mount Sinai COVID Intelligence Center to a more general clinical intelligence center to not just address the needs of COVID, but address the needs more generally of us being able to combine research with clinical care more efficiently to benefit patients. It's a genuine pleasure and honor to be part of this annual meeting.
So we welcome you back. We've just had a momentary interlude as we've been connecting to Professor Cho in New York for the question and answer session. We're just checking that, confirming that she's online.
Yes, I'm online.
Right. So Professor Cho, my name is Warwick Britton. Thank you very much for being with us and for that fantastic talk. Just as an introductory question, as we have a number of questions from around Australia, is the interaction between the clinical research and the Mount Sinai Health System administration is obviously been extremely important to the development of this Biobank data bank. Would you just like to tell us something about how the clinical research and your role as director of the Biobank interacts with the health system itself?
Yes. Probably the biggest interaction is at the level of the entire electronic health system. And so we've had a very close interaction with the CIOs at Mount Sinai. So the entire clinical data warehouse has been made available to the Biobank. And that is kind of by virtue of strong school support, as well as frequent meetings between the CIOs that maintain these electronic health records. Over the years, there's been increasing consolidation. Mount Sinai got into the data warehouse efforts through the consolidated health system early on. And over the years, a myriad of different clinical databases, the cardiac databases, the pathology databases, are all increasingly getting integrated through the Epic data system. So it's very much been a hand-in-hand interaction between the clinical realm as well as the research and biomed. And the final point I'll make is that we've made very active efforts to go throughout the health system. Amanda Merkelson, the managing director of BioMe, is continually interfacing with the major clinical outpatient groups in order to enhance recruitment and enhance relationships.
And this continues to recruit new patients coming into the Mount Sinai Health System. I think you commented that something like 1% of the individuals in the whole health system were involved, but of the 60,000 people in the biobank, is that continuing to increase on a regular basis?
Yes, we're continuing to recruit. Partially, we were stopped for several months due to the COVID pandemic. We restarted. We're watching carefully the small increase that we've seen more recently. We don't anticipate it's going to be nearly as substantial as the peak that we saw in March was, but we're keeping a very close eye on the rates of that. Mount Sinai's instituted. We're much better prepared this time to manage this than we were at the beginning for obvious reasons.
Thank you. Catherine?
Yeah. Can you hear me?
Just come
Yes.
Yeah. Hi, Judy. It's Philip O'Connell. I was interested in I suppose, doing the research and how you take that into the clinical practice. So at one level, it's one thing to look at the genes and look at the mutations, but you still need to have a very good understanding of the biology, because how those mutations interact with other genes may be different, and by just identifying that may not lead to the right therapies. And how are you proposing to do clinical trials in the drug repurposing space? So when you have like 3% or 2% of congestive cardiac failure, for instance, in the case you brought up, have you been thinking about how you do a meaningful clinical trial for a therapy or an intervention you may make with that cohort of patients, as distinct from having a trial where you've got 10,000 people?
Sure. We do have a clinical trials office within the Department of Medicine. In addition, we are working very closely traditionally with the Immunology Institute. And probably like most health systems, the clinical trials infrastructure is extremely well developed in cancer space. And so increasingly over the last, probably decade or so, the interface between the cancer trial space and immunology has intertwined for obvious reasons, with the proliferation of checkpoint inhibitors, the capacity to sequence the tumors. And so there is a strong infrastructure in place in that space. And what we're working with the Immunology Institute is to do small, deep mechanistic studies. In many cases, this is in partnership with pharma. We're increasingly thinking about, and I gave the example of Plaquenil in inflammatory bowel disease, of doing small clinical trials with very deep mechanistic components to them. So the Immune Monitoring Center at Mount Sinai is very accustomed to this because of their cancer experience, and we're trying to extend that to inflammatory bowel disease as well.
Thanks very much. We're now having questions coming from around Australia, and a number of questions are around the application of the PRS, the polygenic ratio score. And one question is that most PRS are constructed from Caucasian data. Do you think such PRS are applicable to non-Caucasians? And that was obviously implicit in your talk. And can you comment on the content validity of the PRS and how you practically apply that?
Yes. So the polygenic risk scores have definitely, because of the ratio, the number of GWAS studies, have been disproportionately balanced in favor of European populations. I think that over the next several years, Far East Asian population PRSs are going to be quite available for researchers to apply. There's very strong data from a number of different traits that the PRSs don't correlate well across populations. And I didn't present these data thoroughly, but in BioMe, for example, if we look at the handful of IBD patients, the PRS is actually quite good. If you take the European ancestry polygenic risk scores and apply them to the BioMe population, we do a pretty good job of predicting traits or not in Europeans. We do a very poor job of predicting in African American cases of inflammatory bowel disease. And I think that reflects both the differences in the case reference size, with the reference case control cohorts being substantially larger in European ancestry populations compared to Africans. And so a major effort within the Inflammatory Bowel Disease Genetics Consortium is to try to, we have an audacious goal of doubling or even tripling the size of the African American case control cohorts. And we have some data to suggest that if you actually use the African American case control cohorts and develop a polygenic risk score that incorporates both European as well as African reference case control cohorts, you actually improve prediction in European populations as well. And again, we can speculate that's due to the fact that you have shorter linkage disequilibrium blocks in African American populations. And the final thing I'll say is that increasingly there's new single cell technologies that we haven't applied yet, but is out there and available commercially now, of single cell ATAC-seq, which will allow us to understand what the transcriptionally active regions are at the individual cell level. And in the case of inflammatory bowel disease, for example, we can actually get a very complete atlasing of that. So I think that in general, a major goal for the field generally should be, for all traits, really increasing the number of African populations. I think that's of highest priority.
Thanks very much. There are some further questions, particularly about the Biobank data bank, in terms of the access to the data bank for international collaborators, and the and particularly, I guess, following on your comments about other groups, particularly Asian groups as well as African American groups. What is the is there a mechanism for access? And how could international researchers collaborate with the data bank? Just to provide a background from Australia, for instance, in Sydney, there's an extensive study called the 45 Up study, looking at a predictive population over the age of 45. But that has been collected in addition to the health service. It's not part of the health service. So the availability and the collaboration.
Yes. So the Mount Sinai BioMe has collaborated very actively on a number of studies. And the mechanism of this, internally with the Mount Sinai, we make the BioMe data available to all Mount Sinai faculty. Beyond that, the major mechanism has been through collaboration. And there are a number of very active groups within Mount Sinai that would be happy to collaborate. We don't have the infrastructure of, say, UK Biobank, where, I mean, UK Biobank is an amazing resource that we utilize and is really kind of an international gem. We don't have that mechanism for sharing. It is typically through collaboration with Mount Sinai investigators, which again has been very successful, and there have been many, many, many collaborations and publications from that.
Okay, thank you. So the question, I guess, relating to the therapeutic targets which developed in this process. I think you gave us an excellent example of repurposing of drugs. And there are emerging targeted therapies. One can think in cystic fibrosis in particular of targeted genetic therapies. One of the issues, of course, is those developed therapies are extremely expensive. And it's the balance of repurposing drugs, which may increase accessibility to the therapies, as opposed to the intellectually and technically challenging but development of new drugs and the availability of those. Would you like to comment on balancing that in delivery of care to the health system?
No, I mean, it's a very challenging question because, I mean, the costs for tafamidis, I don't actually have it off the top of my head, are quite daunting. So you make that diagnosis of precision and the cost of like hundreds of thousands of dollars per year in order to do that is daunting. And so in terms of repurposing, I do think that there are substantial benefits to repurposing. Have in the case of Plaquenil, obviously you have to be careful and you have to check for retinopathy and so forth, but you have a very long track record of safety, which is substantial. The development of these kind of targeted therapies is very expensive. I don't purport to be an expert in understanding the drug economics of this, but I think that increasingly in the field of inflammatory bowel disease, kind of biologic development has been incredibly successful. And I do think that over time, kind of the different targets are going to be very substantial. And I do think that at some point it's inevitable. The cost benefit relationship is going to drive healthcare decisions.
I'll just introduce Chris Samsarian, who will be speaking subsequently.
Thank you for a wonderful talk. I just want to ask a question specifically related to the translation of therapies in the precision medicine field. Specifically, you gave an example of amyloid cardiomyopathy, for example, and a targeted therapy. Who should be delivering that model of care? Should it be the cardiologist? Should it be the geneticist? Should it be all of the above? How do we deliver precision medicine in your view?
Yeah, no, that's a great question. I think that, and I don't purport to say that we've answered that question. I think what's increasingly going to be happening is that the subspecialists, the new generation of subspecialists are going to be driving this, that you certainly will not need to have a medical genetics training in order to do this. I think it's going to be driven still. I just can't see a reorganization of healthcare delivery away from the subspecialization model. And so we're trying to do continuing education for our healthcare providers. I think it's going to be increasingly, for example, we have a new program within internal medicine where the new trainees can actually be trained in medical genetics efforts. And so I think I just practically speaking can't see it's going to be driven by the subspecialists, but the subspecialists that have had experience in their training period towards some of these principles. And the final thing I'll say is that there's clearly going to be a massive kind of data analytic portion to this. And there's going to be early adopters and they can disseminate it throughout kind of their subspecialty area. That's kind of what I'm guessing is going to be the model over the next 10 years, but that's just a guess.
Thank you very much.
Judy, thank you for a wonderful talk. Roger Riddell. The introduction of precision medicine has obviously required the cooperation of the health system itself. And I just wondered if you could comment on what are the drivers from the point of view of the health system to go down this path?
Well, I gave one slide on pharmacogenetics, and we're piloting this. The idea that that every single person who enters a health system may at some point have a favorable or unfavorable kind of drug genetic interaction. And so in terms of early adoption of genetics into health systems, can you do it at scale? Can you do it in a way that the traditional medical approach where you're mapping a patient complaint or a patient issue with kind of a test and a reaction to that test, but instead can actually have a more proactive approach to this where you're doing a comprehensive pharmacogenetic panel and that's actually valuable and relevant and meaningful for years, and it can actually help that patient. And so one of the drivers could potentially be decreasing costs that we don't, and again, as a follow-up to the kind of cost question for pharmacy, there's a natural tendency to always want the latest, greatest, most expensive medication, but sometimes some of the older medications that may be affiliated with side effects, bleeding, but if you know that a person doesn't have that particular pharmacogenic variant, you can potentially give the older drug and be assured that they're going to have a better side effect profile. So we hope, again, the pilot is we've done smaller pilot studies. We're going to do a more systematic pilot study that Aniwa and the pharmacy department is going to be initiating, whereby we're just going to test to see where it may be of benefit. But again, issues like, where does the information reside? How long will the individual be within a particular health system? These are all kind of practicalities, but I think that pharmacogenetics is an early in. And then the final point I'll make in terms of inflammatory bowel disease is that, again, there's some concern about whether or not the particular genetic variants kind of correspond to particular medical therapy. But pharmacogenetics is actually another perhaps early and easy entree point, an early driver, that particular HLA associations associated with antibodies to anti-TNF can actually confer risk for development of antibodies against anti-TNF, reducing its efficacy. So I'm thinking that's going to be a very early driver, is cost. And it's something that patients actually feel very good about. And it's not so much that, okay, something horrible is going to happen to me down the line, but rather that these commonly used medications, it's better to be proactive and know about this. So I think pharmacogenetics will be an early driver and early in.
Thank you very much.
Judy, Tony Cunningham, thanks very much for a wonderful initiation to our conference. I think a number of the members of our audience may be interested in what Mount Sinai is doing in the mental health space as far as precision medicine is concerned. Could you just give us a brief thumbnail on that? Thank you very much.
So the CEO of the health system, the dean, are both psychiatrists. So it's an extremely high priority. It's certainly in pharmacogenetic space, the SSRAs, there are kind of pharmacogenic variants that can play a role in that. And so it is a high priority. In terms of the dean, for example, is very interested in kind of the treatment algorithms of identifying kind of SSRA resistant patients, perhaps being more responsive to ketamine. So there's been a very big interest in priority in that. Have we gotten to the point where we can take genetics and then predict that? I think most people at Sinai would probably say no. We've not really delved in that space yet.
Thank you.
Well, thank you again, Professor Cho. That's been a wonderful start to our conference.